Welcome to BioVivacious. BioVivacious is a YouTube channel dedicated to clear fundamental concepts of biosciences. Today we are going to discuss about coiled coil, which is a structure seen in proteins, especially in the fibrous proteins. What is going to be the learning outcome of this particular session? We will learn first of all about how the alpha helical structure is formed. Then we will learn about the concept of coiled coil. We will be talking about the leucine zipper. Then we will talk about the main functions of this leucine zipper. So these are going to be the broad outline of this particular session. Let us understand how alpha helical structure is formed. In an earlier video, we have dealt elaborately with the formation of alpha helical structure. So we have seen that there are 3.6 amino acids in one turn. That makes it 360 degree, which means if the angle between two amino acids is 360 divided by 3.6, that is equal to 100 degree, which will give a pitch of about 5.4 nanometer. When it comes to a coiled coil structure, remember what happens in a coiled coil structure is if the two right-handed helices will come together in order to form a slightly twisted left-handed helices. In other words, in a coiled coil structure, if the whole structure, if the helical structure is slightly compressed, we have seen that 3.6 amino acids are there in one round, one helical structure. So in order to get a full number, you know that you need to have 5 such structures that will make it 18 amino acid. So 18 amino acid will give you a full number, or you need 5 such ring structures in order to make it full numbered rings. If you want to make this with a coiled coil, we have seen it is slightly twisted. That is 3.5 amino acids are only required in order to make one coil. Which means in order to get a full number, that is very easily we can multiply by 2 and we will get 7. 7 is hepta. You will see that in a coiled coil structure, this heptad is being repeated again and again and again. That is the speciality of a coiled coil structure. So let us see how this particular coiled coil structure is formed. So in order to understand that, so let us draw the 7 residues. We will begin from here. This is residue number A, and the bond angle now 360 divided by 3.5 amino acid. It is about 102 degree. So in a circle, it is easy that if you draw a circle, okay. So this will be B 102 degrees. B will give, C will be here. Okay, C 100 C, D will be here. Now this is E will be here, F will be here, and G will be here. So 7 amino acids are arranged. This forms one coil, okay, one alpha helical structure. Now similarly we can also draw another alpha helical structure. What is important for us to understand is individual alpha helical structures are highly unstable in a cell, in a liquid medium, because it has to ensure it is a hydrophilic environment. Now this coiled coil becomes very unstable in a hydrophilic environment. In order to make it stable, what two alpha helical structure will do is they will come together. They will come together and they will minimize the surface area which is exposed to a hydrophilic environment. Therefore, let us also draw one more alpha helical structure. This is helix number 1. This is helix number 2. They are coming together. So the 7 residues can be shown like this. Let us assume here is A, here is B, here is C, here is D, E, F, G. Okay, so we have arranged in the 7. So let us also A to B to C to D, E, F and G. In the 7 residues are arranged. What is important for us to remember? This region is going to be hydrophobic in region. This region is going to be hydrophobic, and this side will be hydrophilic. What is the kind of association that is established? You will see that if the residue number D is very significant. Residue number D, which is, it will be repeated again and again in this kind of a coiled coil structure. And that residue is normally, D is normally a leucine. You know the nature of leucine. It has got a bulky R group. So normally this is a bulky R group, a leucine residue, and A is also normally a hydrophobic amino acid. In other words, this region becomes hydrophobic in nature, and residues number G and E, these residues, they normally form ionic bonds, which means they will have opposite charges present in them, especially G and E. Residues number this B, F, C, G, C, F, B, C, F, they are normally hydrophilic in nature. So B, C, F residues are hydrophilic in nature. They are in contact with water. This is how this structure is maintained. The coiled coil structure is maintained in a cell. You see how stability is established in a coiled coil structure. Therefore, you can correlate this with wherever there are fibrous proteins. It can be in keratin, it can be in silk, it can be in collagen. Wherever there are fibrous protein, stability can be maintained by this kind of an arrangement. What is important for us to see is how exactly this will fit in. In order to understand the fitting in of these residues, we also need to speak about the repeat which is seen, and that repeat is what we call it a heptad. A heptad is if the 7th residue, every 7th residue is going to be a leucine. Since every 7th residue is going to be a leucine, it is also known as leucine repeat. So leucine zipper is a short sequences. It is an alpha helical structure. Now this alpha helical structure, there will be 2 alpha helical structures and they will cross over. Now it will look more or less like the shape of an X. This is how this structure will look like. What is important is these structures have got 2 domains. One domain that will bind with the DNA structure. Therefore, you will find this within the structure of several oncogenes. You will find this associated with oncogene proteins. Now, they are able to bind with the DNA molecules. You will find therefore a binding site for DNA binding and as well as a domain which is rich in leucine. if the leucine normally project outward, this is where if the leucine will project because it has got a bulky R group. Here also leucine, bulky R group. You can consider if this is a leucine, alternately they will be arranged, and that is how the leucine zipper is formed. This is, assume this is the D we have seen in the helical. This will be A, D, A, and this will be D, A, and this is how they will be arranged in the leucine zipper structure. This is the primary knowledge that we need to know about leucine zipper. Now we are going to understand how exactly they will fit in. Because it is a helical structure. Two helical structure, it should not repel. They should come closer and they have to fit together. If you design an alpha helical structure, you will see that if the side groups are arranged, the R groups they will always project out of the alpha helical structure. Let us try to look at the alpha helical structure from top. When you look from top, you will see that when you look from top, the R groups are projecting outward. Now, in an alpha helical structure, you can have either bonds forming between the first residue with the fourth amino acid, and as well as first residue with the third amino acid. So you can have I plus 3 and I plus 4. So these are the two possibilities we are going to look at today. When it is I plus 3, it will have a different bond angle. When it is I plus 4, it will have another different bond angle at which the residues will project out. Let us look at a picture now. We spoke about the linkage between the first amino acid with the third amino acid as well as with the fourth amino acid. You will see that it is forming a helical structure in this manner, and you will see there is a lot of similarity, especially when it is when the amino acids are linked with the first one with the third as well as with the first one with the fourth one. What is the similarity? And as I mentioned earlier, when you look from top, you will find that there is the ridges project outward at an angle of 25 degree when it is I plus 4. At 25 degree, you will see that this is the angle it is taking. And when I plus 3, if the ridges takes a different angle altogether, it will take this is the way it will form ridges. And that is about 45 degree, it forms an angle. So therefore the ridges are projected outward at different angles. What is the significance of this? If the significance of this is especially when you have when you want to fit them together. So in order to understand that, let us draw two. So let us clean this portion. So here is an example of 1 plus 4 is drawn here. 1 plus 4, one helix. This is one helix. Similarly, we have a second helix. This is also 1 plus 4. You will see they are exactly matching. 1 plus 4, second helix is formed. Now what we are going to do is, we will flip one of the helices by 180 degree. So this helix is flipped 180 degree, and you will see that it is not matching and it is overlapping the two helices. Now in order to make these helices stable, we need to fit the ridges into the grooves that is formed in the alpha helical structure. How do we fit the ridges into the grooves? What we are going to do is, in this case, this is overlapping, we will rotate this about almost 50 degree we will rotate. We are rotating about 50 degree. By rotating 50 degree, this has matched. You can see that the ridges etc. has matched into the grooves of the next alpha helix. In other words, with this kind of a formation, with such a formation, it forms a tight, stable structure. This is when we are arranging the alpha helical in the anti-parallel shape. We can also arrange them into a parallel shape. So, in a parallel shape, here is one helix, here is one helix, here is the next helix. Okay, two helices are arranged. Now, these two helices, one is flipped over the other 180 degree. You can see that they are not matching. Now, in order to match these, what has to be done? It has to be rotated. It has to be rotated slightly, about 18-degree rotation must occur in order to fit it. When you rotate 18 degree, the ridges will fit into the grooves and the structure becomes highly stable. This is how the alpha helical structure fits, especially in the coiled coil. We have looked at the structure. Now this structure has given rise to a very famous hypothesis known as PV hypothesis. What exactly is PV hypothesis? It stands for peptide Velcro hypothesis. You understand what is a Velcro? Velcro is something which we use for sticking them together. You will see that, like a Velcro, how exactly one alpha helix fits into the other. One ridge fits into the groove of the other and gives stability to the coiled coil structure. In order to meet a particular function, how the structure has been modified especially by using these leucine zippers. Just now we have looked at the two helical structures, which is forming a coiled coil, which is generally found in nature. And this hypothesis was proposed by, or this concept was given by Francis Crick in 1953 when he was looking at the protein structure. You will also find four helical structures that is found in protein. A four helical structure which will look very similar to this. You will have one helical structure, the second helical structure, third one, and four helical structures. And these are all alpha helical structures. What is important here is this portion is going to be hydrophobic in nature. So this is hydrophobic and the surrounding will be hydrophilic in nature. That is how the four helical structures exist in protein. And these are also stabilized. You will find this kind of coiled coil structure in many proteins. They are involved in varieties of functions. First of all, they are involved in giving stability to protein. They are involved, especially in the soluble proteins, they are involved in signal transduction. There are a number of factors we need to understand about the coiled coil structure. Number one, the formation is pH dependent, that we have seen it. Number two, the structures can be modified by phosphorylation and dephosphorylation because there will be charge separation that can modify the protein. They have varieties of functions to perform. Apart from giving stability to structures, they are involved in molecular recognition. They are involved in signal transduction. Similarly, there are varieties of functions these coiled coil structures performs in living system. What are the things that we have seen so far about the helices? We have seen that formation of helices is pH dependent. We have seen that a helix has got its own polarity. All the amino terminus is looking in one direction, all the carboxyl ends are looking at another direction. Therefore, the polarity we speak about is from the N terminus to the C terminus. The N terminus will have a positive charge plus 0.2. The C terminus will have a negative charge, that is minus 0.42. that is the charge at the C terminus. You can see there is a very clear demarcation in terms of polarity. You have also seen that what drives the formation of an alpha helical structure is the bond angle that is forming between two amino acids drives the formation of an alpha helical structure. You have seen that there are 3.6 amino acids that are present in one helical structure. You have also seen that when it becomes a coiled coil, it begins to compress. If the pitch becomes slightly lesser than a normal alpha helical structure with the 3.6 amino acid. So in the case of a coiled coil structure, if the pitch becomes 5.1 nanometer, slightly become compressed, and that is how it is able to form a stable structure. Stability is also involved because if the ridges that are formed in one of the helices fits into the grooves of another helix and it becomes an amphipathic structure, gives stability. What is important for us to remember is these two structures, alpha helical structures as well as the beta pleated structures, are very common in a protein. Wherever a protein has to communicate mobility, mechanical motion, signaling, that time it will use, generally it employs an alpha helical domain where there is a lot more of stability in the structure, where there is a binding occurs and modification has to happen. That time the beta helical structure is used by a protein. You see how a particular shape or a structure suits a particular function in a protein. So this is how we unravel in the different forms of a protein. I am sure you have enjoyed going through these videos. Keep subscribing and keep watching.